I'm Rainer Benowitz, and welcome to this brief overview of the major research directions, projects, and current collaborations in our research group, the URBS-Lab. Some quick background on myself. My original undergraduate and master's training was in biomedical engineering. Later, my PhD and postdoctoral work was focused on genetics, computational biology, and informatics, with a particular focus on machine learning and its biomedical application. My previous positions have included teaching faculty at Bard College and an assistant professorship at the University of Pennsylvania. Currently, I'm an assistant professor, title pending, of computational biomedicine at the Cedars Sinai Medical Center, as well as remaining an adjunct professor of informatics at the University of Pennsylvania. In the broadest sense, my research lab focuses on the development and application of methods to analyze and seek to tackle the many challenges of biomedical data. There are many possible sources of biomedical data that investigators may gather or interrogate in their research or patient care. For example, this data could come from electronic health records or from genetic sequencing. Data sources such as these are typically structured and can be represented as tabular data, which many computer programs or algorithms can work with directly. As seen here, tabular data is typically represented as rows of instances, which could be patients or other individual data samples, and columns of features, which are the variables or measures recorded for a given instance. Also, typically there is some outcome, or the dependent variable that we're trying to better understand or predict. Generally, we can work with tabular data directly, or apply human-driven feature engineering or machine-driven feature learning to come up with more informative ways of representing the original data for downstream analysis. However, other sources of data, specifically unstructured data, can be more challenging to work with and requires additional steps or specialized methodologies to analyze. For example, text from doctor's notes, images such as MRI scans or histology slides, and time series data, which is data that includes the dimension of time, such as heart rate or EKG data. For these types of data, we can employ feature extraction, conducted either manually by an expert or with the help of specialized machine learning approaches, to extract and assemble tabular data from these unstructured data. Notably, some machine learning methods are designed to work directly with these unstructured data types. To date, most of the research in the URBS-Lab focuses on methods designed to work with tabular data, and we work with collaborators with their own biomedical research questions to access and analyze data once it's in its structured format. However, the challenges involved in feature extraction, as well as working directly with unstructured data, is of future interest to our research group as well. While our focus on tabular data narrows the scope of our lab's methodological work, our research seeks to simultaneously address a wide range of challenges that are characteristic to working with many forms of biomedical data and beyond. Some of the basic challenges include the presence of missing data, imbalanced data, having big or large-scale data to manage, and the need to integrate and analyze a diversity of data and feature types. Consider the wide range of potential biomedical data sources investigators may wish to interrogate in the study of their outcomes of interest. Data could come from the genome, epigenome, proteome, microbiome, electronic health records, imaging, mobile health, and even social media. For some or possibly many problems of interest, it may be necessary to combine these data sources in order to effectively identify risk factors and make predictions. This presents us with a number of logistical and methodological challenges to overcome. Also, when it comes to detecting the relationships in data, biomedical data is often noisy, it holds the possibility of rare variants having an important effect, and of primary interest to the URBS-Lab, there can be complex, multivariate associations connecting risk factors to outcome, making these relationships more difficult to identify. In this figure, the left box represents features in the data that may be risk factors, while the box on the right represents outcomes or phenotypes we want to understand or predict. Relationships can be simple and univariate, with a one-to-one association. They can also be pleiotropic, where one feature can impact multiple outcomes. They can also be epistatic, where multiple features interact with one another in a complex manner to impact some outcome, where these individual features may or may not be associated with outcome on their own. Also, there can be heterogeneous associations, where different features impact outcome in different subgroups of instances. And lastly, there could be some complex combination with any of these relationships. To date, the URBS-Lab has focused in particular on developing methods that are sensitive to and allow us to characterize both epistatic and heterogeneous associations. Beyond the challenges mentioned so far, further analytical challenges regarding interpretation include considering covariates, where we want to understand the impact of covariates with the goal of separating potential causal relationships from the associations found in data. We also ideally want our models and results in general to be transparent and understandable, or in other words, we want to avoid black box decision making, so that researchers, clinicians, and patients alike can trust the predictions and decisions made by machines. And furthermore, we want our results to be reproducible, both in terms of how our methods work and in terms of the reproducibility of the biological findings. To introduce the methods our lab develops, let's first define and distinguish the terms artificial intelligence and machine learning. One definition of artificial intelligence, or AI, is getting computers to do tasks that require human intelligence. This is a very broad definition, but largely captures what AI focuses on. In our lab, the basic element of AI that we're most interested in is automation, specifically towards improving the speed, efficiency, and ease of use of analytical tools. On the other hand, machine learning is defined as a subset of artificial intelligence in the field of computer science that often uses statistical techniques to give computers the ability to learn with data without having been explicitly programmed. Here, our lab focuses on leveraging machine learning to perform analysis at a scale humans or existing methods can't achieve. We also leverage these methods to be able to discover novel patterns in data and identify risk factors that other simpler methods might miss. However, our lab's focus on machine learning isn't just about the algorithms themselves, but how to apply these methods as part of a holistic data science pipeline to identify best practices, avoid bias, ensure reproducibility, and enhance transparency and comprehension of the results as a whole or individual predictions made by modeling algorithms. Now let's take a big picture look at some of the machine learning and artificial intelligence research themes in the URBS-Lab. With respect to AI, our interest is in developing tools to automate machine learning and data analysis as a whole. This subfield of machine learning is also referred to as AutoML, or automated machine learning. Here we're trying to answer questions like, how can we make machine learning easier to use, more effective, faster, less biased, reproducible, and easily comparable to other state-of-the-art methodologies? Also, how can we make machine learning more adaptable to different datasets or problems, and at the same time make fewer assumptions about our data going into the analysis? With respect to machine learning, our lab is interested in developing new methods and algorithms to address many of these challenges we've reviewed in data science. Here we're trying to answer questions like, what are the strengths and weaknesses of different machine learning algorithms in different contexts? How can we improve methods for quickly, effectively, and interpretably detecting feature interactions, heterogeneous associations, or additive associations? As well as how do we detect the impact of rare variants? Further, how do we scale existing or new methodologies to big and integrated datasets? And furthermore, how do we develop or identify benchmarking data or benchmarking approaches to test or compare methods? And of course, application of new and existing methods to a variety of datasets or problems in medicine or biomedical research. Now I'm going to briefly introduce you to some of the key algorithms, software, or tools developed and in development within the URBS-Lab. Currently, the central hub of our research program is our automated machine learning pipeline tool called STREAMLINE. This project focuses on making machine learning easy to use, reproducible, and interpretable. Next, we have our software GAMETES, a program for simulating complex genetic data. This software allows us to create controlled, yet diverse and complex simulated data to test, evaluate, and compare our machine learning methodologies. Another area of research focus involves feature selection. Our software package ReBATE implements a variety of interaction-sensitive feature selection approaches, allowing users to conduct feature selection in a manner that prioritizes features in the data that are involved in an interaction. Another major focus is on the development of machine learning modeling algorithms themselves. I've developed a strong interest in rule-based machine learning modeling, and we've previously developed ExSTRACS, a rule-based machine learning algorithm designed for learning on biomedical data. The reason I'm so interested in rule-based modeling is that this methodology pairs interpretability with the ability to detect extremely complex patterns in data, including epistatic interactions and heterogeneous associations, as well as provides an avenue to directly detect and characterize heterogeneous subgroups in data. Our lab currently has 3 scikit-learn compatible packages out, allowing users to use ExSTRACS and 2 other well-known rule-based machine learning algorithms. Furthermore, In order to facilitate the interpretation of the rule-based machine learning output, we've recently developed LCS-Dive, an automated rule interpretation package that facilitates the detection of heterogeneous subgroups. And the last major project I'll mention is also the newest. Fibers is an automatic feature learning tool that uses an evolutionary algorithm to automatically identify bins of features in the data in a way that combines them in a maximally informative manner. All of these tools are currently available for use and are being actively improved and expanded upon in our current research. For the remainder of this research overview, I'm going to quickly provide a little bit more detail on each. First, I'll start with STREAMLINE, the current hub of our research program. STREAMLINE is an automated machine learning analysis pipeline, currently set up only to accommodate binary classification problems. But we're currently expanding to multi-class and regression outcomes as well. STREAMLINE is an end-to-end pipeline for conducting machine learning predictive modeling in a rigorous and transparent manner. When I say that it's end-to-end, I mean that it automates all of the following elements: exploratory analysis, data processing, feature processing, modeling, analysis summary, significance analysis, figure generation, and it even generates a PDF report. We designed STREAMLINE to adopt current best practices in machine learning analysis, and it offers flexibility, customizability, expandability, and reproducibility to its users and open source developers. We focused on designing it to be able to tackle the following major challenges, including dealing with common data challenges like missing data or class imbalance, scaling its application to bigger data analyses, and ensuring that the pipeline was set up to be able to detect and interpret complex associations in data. You can download and read more about STREAMLINE at the GitHub link here. Here's a more detailed schematic of everything that STREAMLINE currently includes. I'll leave the details to another video, but I'll highlight the major phases of the algorithm that includes data preparation, feature importance and selection, modeling itself including 15 different machine learning algorithms, and post-analysis, where the results are summarized, organized, and statistical significance analyses are conducted. The pipeline is also set up to easily allow users to rerun all the models that they've trained on some new external data in the future. To illustrate why STREAMLINE is currently the hub of our research program, our data simulation tool GAMETES allows us to generate different data types to conduct machine learning experiments. Furthermore, our feature selection software ReBATE is integrated into the STREAMLINE pipeline, and the rule-based machine learning algorithms we developed, including ExSTRACS, have also been integrated alongside other well-known machine learning algorithms that have been added to the STREAMLINE framework. In addition to making it easy to run a comprehensive machine learning analysis, STREAMLINE also outputs a wide variety of publication-ready figures. This includes results from the exploratory analysis, such as plots of class imbalance, univariate analysis, and feature correlations. And after feature selection, it generates estimated feature importance rankings prior to machine learning. During machine learning optimization, it outputs illustrations of the hyperparameter sweep used to optimize each machine learning algorithm. Following modeling, it outputs ROC and PRC curves comparing all machine learning algorithm performances, and plots of estimated feature importance as identified by each individual machine learning algorithm. For some machine learning algorithms that are considered to be interpretable, such as decision trees and genetic programming, STREAMLINE also allows the user to directly generate visualizations of those models. And if the user chose to run multiple datasets in STREAMLINE simultaneously, the software will also automatically compare overall performance between these different datasets. And lastly, as mentioned before, STREAMLINE creates a PDF report summarizing critical information about the dataset, the run settings of STREAMLINE itself, and key tables and figures highlighting the results of the analysis. Moving on, I'll briefly mention GAMETES. This simple software allows users to generate complex datasets with epistatic, heterogeneous, or additive associations in data. It can be run from the command line or in this simple graphical user interface. If you want to read more about GAMETES, check out any of these 3 publications dealing with data simulation and the GAMETES software. To download or read more about the GAMETES software, check out this link on GitHub. Moving on to ReBATE, which is our software for interaction-sensitive feature selection, this scikit-learn-compatible software package allows users to easily run any of a variety of relief-based feature selection algorithms, including our own top-performing feature selection algorithm, MultiSurf, which is currently incorporated into STREAMLINE. Our previous research demonstrated the efficacy of these core feature selection algorithms, but also identified that when the feature space gets particularly large, their reliability in being able to detect pure epistatic interactions begins to degrade. As such, our lab is also interested in wrapper approaches and other ways to improve the performance of these relief-based algorithms. If you're interested in reading more, check out this review paper on relief-based feature selection algorithms, and our research paper introducing the ReBATE software and MultiSurf. If you want to download scikit ReBATE, check out this GitHub link here. In line with this work on interaction-sensitive feature selection, we've also developed an interaction-sensitive gene set enrichment analysis approach that integrates ReBATE's relief-based algorithms with the traditional gene set enrichment analysis strategy. We applied this approach to the analysis of genetic risk factors in congenital heart disease, and demonstrated that we could gain more insight using gene set enrichment analysis when we took interactions into consideration. The last thing I'll mention about ReBATE and its incorporation into STREAMLINE, our AutoML tool, is that STREAMLINE combines our interaction-sensitive feature selection method with a more traditional mutual information-based feature ranking approach. And we combine these methods when we conduct feature selection using a collective feature selection approach, as pioneered in this publication. Moving into the topic of machine learning modeling, you may already be aware that there are many different supervised machine learning algorithms out there. This slide highlights a variety of families of algorithms, along with specific algorithms themselves. Among these are rule-based machine learning algorithms, and in particular, learning classifier system algorithms, which is the basis for our own lab's rule-based machine learning methods. As a quick introduction to rule-based machine learning, this is an alternative paradigm of modeling, which seeks to model a problem space in a piecewise manner using a set of rules. So for example, let's say we have a classification task where we're trying to distinguish our blue circles from red X's. These could represent healthy and sick patients. Rule-based algorithms learn individual rules that each cover a subset of the problem space. Here, each blue or red box identifies a rule that has been learned to predict one class or the other based on the problem space that respective rule covers. And at the end of learning, the set of rules as a whole determines our class prediction boundaries. So what is a rule? A rule is just an if-then expression that captures a feature to outcome mapping. Here are some rules that might have been trained on a dataset including genetic variants, such as SNP-A or SNP-X, and other clinical variables, such as BMI or body mass index. If you want to learn more about rule-based machine learning, and in particular learning classifier systems, check out our brief animated video called Learning Classifier Systems in a Nutshell. And if you want to learn even more, myself and collaborator Dr. Will Brown previously wrote a textbook called Introduction to Learning Classifier Systems. This book pairs with a simple implementation of a learning classifier system that we called ELCS, or Educational Learning Classifier System. This implementation is also currently available on our GitHub page. Some of my early research aimed at demonstrating their efficacy at detecting complex biomedical patterns as a proof of principle. Later, we developed strategies for statistical analysis and interpretation of the rule sets that are trained by these algorithms. Further work looked at how we could take existing expert knowledge, either from the biomedical domain or from prior statistical analysis, and use this knowledge to guide the rule-based machine learning such that it could more optimally detect these complex patterns. In this work, we showed how feature importance estimates being generated by our interaction-sensitive feature selection method in ReBATE could accelerate learning in a rule-based machine learning algorithm. Furthermore, we developed a novel strategy to directly characterize heterogeneous subgroups in data from rule-based machine learning. Specifically, we developed something we called attribute tracking, and we applied this to successfully identify patterns of heterogeneity and epistasis in bladder cancer. Taking a quick closer look at attribute tracking, during the course of these learning classifier system algorithm trainings, for every individual instance in the dataset, the algorithm is able to collectively track the importance of each feature involved in correctly predicting outcome for that individual instance. This concept is inspired by the pheromone trails left by ants in search of food. Here we can see a heat map visualizing these attribute tracking scores following training of the learning classifier system. Cells in yellow identify features that are more important for predicting the outcome of a given instance. And in this current simulated data scenario, we can see that the attribute tracking scores identified two major subgroups, each characterized by a unique epistatic interaction between two features. We took everything we learned and developed from this initial research, and we used it to build ExSTRACS, our own rule-based machine learning modeling algorithm developed specifically to handle the challenges of biomedical data. Here we have the first two publications on the ExSTRACS algorithm, where we focused in particular on scaling these approaches up to larger datasets. And at the same time, we were the first ever to directly solve the long-standing computer science benchmark problem known as the 135-bit multiplexer problem. While this is just a toy problem, it critically illustrates how rule-based algorithms can not only detect extraordinarily complex patterns in data, but can do so in a way where the model is represented with a collection of simple and interpretable if-then rules. We've taken this research in rule-based machine learning in some other directions as well. We've also explored ways to get rule-based machine learning to automatically adapt to different levels of data complexity, such that they can perform better without making any prior assumptions about the data. We've also worked on ways to improve our aforementioned attribute tracking strategy, and ways to better adapt rule-based machine learning to quantitative outcomes. And since this family of algorithms isn't well known, and there isn't much in terms of accessible software, we've turned ExSTRACS and the other learning classifier systems mentioned into scikit-learn compatible software packages that are open source and easy to use. You can find these packages at the GitHub links at the bottom of the page. Probably one of the most unique focuses of methodological development in the URBS-Lab is on the ability to detect and characterize heterogeneous patterns of association. Recently, we published a review paper covering the challenges, impacts, and methods used to tackle genetic heterogeneity. And as mentioned, we're actively developing LCS-Dive, an automated pipeline to take the output of ExSTRACS, including the rules and attribute tracking scores, and automatically identify heterogeneous subgroups in data, where different subgroups have different single or combinations of risk factors that predicted their outcome. You can check out LCS-Dive at the GitHub link here. And the last piece of software we'll take a closer look at is Fibers, which focuses on feature learning and rare variant analysis using binning. The Fibers algorithm is an immediate descendant of the RARE algorithm we previously developed. RARE was designed to identify bins of features that together improve the association with some binary outcome. Fibers expands on this idea and adapts it to survival data, where we have some time to event of interest. In Fibers, bins are discovered and evaluated such that they maximize the difference in survival curves of the risk groups defined by the bin itself. This algorithm was applied to risk stratification of kidney graft failure, focusing on HLA amino acid mismatches as the features, and time to graft failure as the outcome. In this recently submitted work, we showed that the top amino acid mismatch bin discovered by Fibers yielded similar risk stratification in comparison to traditional antigen-level mismatching. However, the Fibers bin was able to stratify a much larger proportion of potential transplants as being low risk. This preliminary work was recently funded as a new R01 and will be one of the focuses of our lab research for the next 5 years. I'll end this overview by reviewing some of the major collaborations and funding driving our research as of January 1st, 2023. As just mentioned, the Fibers project is focused on donor-recipient kidney transplantation, and this work was recently funded as an R01. This work is being conducted in close collaboration with Malik Kamoun of the University of Pennsylvania and Lauren Gragert at Tulane. In this work, the URBS-Lab will focus on the further development of Fibers and other machine learning methods and their application to this transplantation research. We're also currently working in the domain of sleep health, including obstructive sleep apnea and insomnia. This work is in collaboration with Alan Pack and Brendan Keenan of UPenn, as well as Diego Mazzotti of the University of Kansas and Ulysses Mangalan at Ohio State. This research focuses on the identification of risk factors, the characterization of heterogeneous patterns of association, and the many challenges of data integration. Furthermore, we'll be collaborating on a recently funded Alzheimer's disease grant, led by Dr. Jason Moore at Cedars Sinai and Marilyn Ritchie and Lee Shen at the University of Pennsylvania. This work will focus on the development of open-source artificial intelligence methods to facilitate Alzheimer's disease research. We're also collaborating in the domain of thrombotic events and readmissions through the analysis of electronic health record data. This is in collaboration with Danielle Mowry and Randall Oyer, also from the University of Pennsylvania. This project combines elements of natural language processing and machine learning for predictive analysis. And our last current major area of collaboration focuses on pancreatic cancer. This collaboration is with Shannon Lynch of the Fox Chase Cancer Center and Rachel Stolzenberg-Solomon at the NIH. At a high level, this project is focused on integrating epidemiology with machine learning in the identification of pancreatic cancer risk factors. Thanks for joining me for this overview of research in the URBS-Lab as of January 1st, 2023. Please feel free to reach out to me if you're interested in collaborating or learning more about our research or the tools and software that our lab has produced.